This is Dr. Wright, and in this third part of normal lung anatomy, we're going to concentrate on the conducting airway system. So our learning objectives are to understand the branching system of the airways, to understand what constitutes the conducting airway compartment, and to understand the gross and microscopic characteristics of the conducting airways. You've seen this cartoon before if you've looked at parts 1 and 2 of the normal lung anatomy series. So first of all, the trachea constitutes generation 0, and then the lungs bifurcate into main stem bronchi, segmental, and subsegmental airways, all the way down to generation 19, which is the terminal membranous bronchiole. From generation 0 to generation 19 constitutes the conducting airway system because it doesn't contribute to the respiratory component. It only conducts the air down to the respiratory component. On this cartoon, you can see the trachea, bifurcation into main stem bronchi, segmental and subsegmental airways, which form the various lobes and lobe segments. So let's start off with the trachea. On the CT scan, here's the more proximal aspect of the trachea, coming down to bifurcate into main stem bronchi, bifurcating again into segmental and subsegmental airways, forming the various compartments of the lung lobe architecture. On this axial section, you can see that there's something different about the posterior aspect of the trachea, and that is that it is formed of fibromuscular tissue rather than the cartilaginous ring, which forms the anterior aspect. In this cartoon, you can see the cartilaginous rings, which extend down into the segmental airways. On this cross section, here is the cartilaginous ring, and in the posterior aspect, it's fibromuscular tissue. The muscle is called the trachealis muscle. Posterior to that, of course, is the esophagus. On this section, it again shows the entire cross-section of the trachea. Here's the cartilaginous ring. Here's the posterior aspect with the trachealis muscle. On this low-power gross micrograph, the hyaline cartilage, the mucous gland lobules, the pits, which allow the mucous glands to exit onto the surface of the epithelium, and the epithelium of the trachea. On a higher magnification, you can see the mucous gland lobules, which are seromucinous in type, the bronchial pits, as you showed before, coming up onto the surface to allow the mucus to come onto the tracheal surface. On a higher magnification again, you can see that the respiratory epithelium is pseudostratified in type, and it is ciliated. It is pseudostratified, and it has a fair number of goblet cells in it, again contributing to the mucus layer which is present on the top of the epithelial surface. So, just to reiterate, the conducting system from generation 0 to generation 19 of the airway system, the conducting airways. This is a smaller subsegmental airway, you can tell because it still has cartilage in it. It shows the respiratory epithelium, the muscle, and the space in between the basement membrane and the muscle, so the subepithelial space. Over here is a lymphoid aggregate, which is quite commonly found adjacent to airways and is part of the immune system. On a higher magnification, again, you can see the association between the bronchi and the vascular system. This is called the bronchovascular bundle, and interestingly enough, the diameter of the airway is very similar to the diameter of its adjacent artery. On a higher magnification again of the epithelium extending down towards generation 17, generation 18, you see it's ciliated, pseudostratified respiratory with a large number of goblet cells. This is an electron micrograph of the cilial surface, showing the elongated cilia extending from the surface of the epithelial cells. This is an axial view of the epithelium of the cilia, which shows that its structure is such that it allows the cilia to beat, and to beat actually in unison, allowing for mucus clearance as it goes to the surface. This is a cartoon which shows that the cilia are embedded within the mucin, allowing their beating to extend the mucus to go up proximally, where you can swallow it or spit it out depending on your inclinations. These are the ciliated epithelial cells, and there are a number of other cells within this epithelium. Some of them are club cells, formerly called Clara cells, which are important cells because they have a large number of antioxidant enzymes within them. All of this forms the mucociliary escalator, which removes the material trapped in the mucus up towards the proximal aspect of the lung. There are neuroendocrine cells within this epithelial surface, and the neuroendocrine cells are important for the tumors which arise from them. They're also important in the physiology of airway constriction. So the learning points of this portion of the lung anatomy series are what constitutes the conducting airway system, the structure of the trachea, the structure of the membranous bronchioles, the types of epithelial cells, and their function. Here are your questions. The answer to question number 1. Here's question number 2. The answer to question number 2. Question number 3. The answer to question number 3. Question number 4. The answer to question number 4. And I hope that you've enjoyed this component of the normal lung anatomy series.